All right, well, thanks. I was really excited to find a conference on Bayesian networks because that's basically what I do. So this is going to be a tag team presentation. I'm going to give a bit of overview of some of the things that we do in my lab and on different biological systems, and then my student Edwin is going to come in and talk about his particular project in ecosystems. So what my group is interested in is the problem of biological network inference. So we've got a system of some kind of variables. So these might be regions in the brain or different species. And they interact in some way, and we want to know that. That's basically the question we want to know. So we apply an algorithm to this to get out the set of putative interactions. You can probably guess the algorithm that we use is Bayesian networks. And in particular, we use heuristic searches for the best Bayesian network in a score-based system, and we've used a number of different scores. And so today I'm going to go through some stuff that we've done in neural systems, genetic systems, and a little bit of ecology. I don't know how much I'll get to because I'll take a look at the time and then jump ahead to Edwin's, who will give us about his work in rocky shore ecosystems and ecological resilience. So starting off with neuronal information flow networks. So this is the thing that we're trying to figure out, and this is the biology behind it. The biology behind it is that you've got neurons in the brain. Each neuron, if it fires an action potential, that action potential will travel down an axon, hit the end of the axon, where neurotransmitters, which are small molecules, are released. They go on, they hit the receptor of another neuron, and that either increases the probability that the next neuron will make an action potential or decreases it. So it's activation and inhibition, and a combination of these is what we call the neural information flow network.
We have to swap that
We have
out.
to swap.
I think this is, the battery's going dead.
Okay, I'm getting quiet there.
Yeah. Perhaps that would be better.
Okay, so one of the reasons, is that working? Sound? Same? Do I need to shift the location? What if I put it up here? Will that help? Is that, whoa, no, that definitely won't help. Over there? No, that's not, it's on.
Is it on?
On, on.
You know, let me, let's do it the old-fashioned way.
I'm gonna hold it this way. All right,
I'm sorry.
there we go. All right, I feel like an MC. Here we go. Got that. All right, how's that? Is that working? Is that good? Is that good?
Yeah.
Okay.
Okay. All right.
Comes and goes.
Comes and goes. Like this. There we go. How's that? Oh, I can hear it. Wow, this is weird. Okay, so neuronal networks are actually really suited to this structured learning and Bayesian networks. We can get huge amounts of data from a neural system, thousands or tens of thousands or more data points that we can collect from an animal. It comes in a time series of one point after another. This enables us to use dynamic Bayesian network models, which enables us to model loops, which biologists care about a lot. And so we can easily have loops by having kind of 2 time slices and getting loops across time, but the Bayesian network is still acyclic. And additionally, neurons have actually been hypothesized to interact with each other in Bayesian manners, kind of like a Bayesian network. So it's a really nice system that we're trying to recover with Bayesian networks. So I'm going to start with just showing you some stuff the first stuff we did with this. This is with Jing Yu when she was at Duke University here, and I was here with her using some data collected from songbirds. This is a female zebrafinch. And the reason we did this is they actually know where all the axons go in the brain in this particular region where they're listening to sound. So we could put electrodes in there, we can try to apply our Bayesian network algorithms to them and see if the structures we recover flow over known paths. And if they do, we're quite good. And basically, the only reason I have this slide is because I want to play you sounds. So we had female birds, and they listened to songs. And this is the song of a male. And we also gave them control sounds of white noise. And then that white noise modulated to sound have the same amplitude envelope as a song. So we had 6 birds. They each got 4 different stimuli on 4 sessions. For each of those different stimuli per session, we were able to recover a Bayesian network. We used the BDE score with discretized data, but we still were using 20,000 data points. And we had 16 networks per bird. We combined those, and we combined each of the birds together to get our, let's see. Oh, I didn't know it was going to do that. Okay. There, okay, that worked. All right, so we combine those together to get a consensus network that we've recovered, and we can compare this to the known anatomy. Well, you may first see that there are a couple links here that we don't have in the known anatomy, but these are actually unknown links. So there was a region that we recorded from that they don't actually know how it's connected. So these links, that there and that there, represent hypotheses about where the neurons should be going, the axons. And there was also some electrodes in the same region of the brain. They don't know the interconnectivity in there. But of the remaining links, they all float over known connections. You may be looking at that and thinking that's a very densely connected network. So could have that just happened by chance? But in fact, we know there's some links that don't exist, and there was also some electrodes in other areas of the brain, and we significantly avoided these. So we're reasonably convinced that what we're getting is a reflection of the neural information flow in the brain. And then we can do things like look at the difference in the neural information flow when they were being played that noise stimuli to a more biologically relevant song stimuli. And here with the noise, you can see this is actually a region where the sound comes in from the nuclei from the ear, and it mostly kind of went from there to this area where it goes out to the rest of the brain. Whereas the song stimuli spent more time. It came from these more higher processing regions out to the rest of the brain. So those were differences in the information flow. Now, continuing to use these, we wanted to apply them to single neurons as opposed to what we had been doing with that songbird, which was just putting an electrode in its brain and measuring kind of several hundred neurons there. But the problem with that was that this data was basically normally distributed, whereas a single neuron is much more sparse. It's more like a Poisson distribution. And in the BDE score we were using, it has a lack of semantics. So basically what was happening is when 2 neurons were quiet, they looked very predictive of each other, and they'd be connected. And that's not what we want. The spikes, those action potentials, those are the things that are making the signal. So I had a PhD student who developed a new score for use with single unit neural data, and where he treated spiking and non-spiking differently and developed a hybrid score where you use the spike of the child node and got a convolution continuous activity level series of the parents. And this is a likelihood score at the moment, and it's just a conditional probability that the child will be spiking given the parent's activity. And then we were able to use this. This is actually, I love this. This is data from blind mice on their retinas. And what the blind mice do is they just have kind of activity waves that go across their retinas because they can't see. So they've just got this random activity that happens, and we could see these waves moving across the retinas. And the last bit here is just kind of our current work. This is the type of data that we're starting to use now, which is imaging data. This is a dissected neural cord of a Drosophila larvae, and it's exhibiting fictive motion. And the brighter things are when neurons are firing. So we're measuring the neurons with light. This is it's called a calcium signal. And I've got some undergraduates who have worked on this and a PhD student, and we're working on identifying regions of interest here and figuring out where the neural information flow is going when these larvae are fictively calling forward or backward, for example. All right, and I think it's actually going to start the movie on me again. There we go. How do I make it go sideways? There, okay. So that was the neural systems. We also work in gene regulatory networks. This is kind of the biology behind this, and this is a much more complex system where you've got your gene in the DNA. This is expressed into messenger RNA that turns into protein. And the protein is the thing that might then go and either make another gene turn into RNA, or make it stop, inhibit it. And the combination of these activators and inhibitors is what we call the gene regulatory network. So when we started off with this, so unlike the neural systems where we had 20,000 data points just quite easily, we actually had 16 times that just from one bird. In gene systems, you have very, very few. This data costs $100 or more per data point. It's very expensive to get. And we did some simulation work looking at kind of the recall, so the amount of a network recovered, and the imprecision, which would be the amount of the answer that we got that was wrong. And you can see in these low, kind of biologically realistic data amounts, we were getting really bad network structures. And so I'm going to talk to you about this challenge here, decreasing the false positives. This, again, was work that was done with Jing at Duke. And she developed a number of features to help tackle this. One was an influence score for a Bayesian network, for a discrete Bayesian network. And you can see how her influence score, so she had a simulated interaction strength, which was producing continuous data, and then the influence score nicely tracked the interaction strength. And what we use this for to decrease those false positives was say, well, if it's really, really, really small, maybe that's an artifact. And yeah, that helped a little bit, but it wasn't a big thing. Another thing was interpolation in time series. So remember, we often do these dynamic Bayesian networks. And I, at first, when she suggested this, thought it wasn't going to work, because it's just making extra data. How is that going to help from your same data set? But in fact, it worked quite well. And we realized what we're doing is we're adding an assumption to the model. We're adding the assumption that we've got two sets of data, and we know what happens in between them. The data should go in between them. The algorithm had no idea about that. And it was really this first sampled data point, this first interpolated data point, that really made a big drop in our imprecision. And finally, we did some theoretical work on the BDE score, finding that if you didn't fill the scoring matrix well enough, you did tend to get a proliferation of false positives. So we identified a parent number that you could limit your structure search to avoid this. And that actually had a really big impact on getting rid of these lots and lots of false positives at low data amount. And here's an example of doing it. This is just kind of an unadvanced dynamic Bayesian network with the BDE score on 36 data points. It's this rat's nest. If we applied all of those, then we came down to kind of 4 interactions. So we got rid of lots of stuff. And I'm not going to talk about, there's some reason to believe some of these could be connected. But one of the things I've discovered working with this is when we started it, we thought we'd like to make these gene regulatory networks. Like, that was our goal, but it turns out it's not really the biologist's goal. What they really want is kind of genes or groups of genes of interest to go ahead and do some more experiments with. So instead, what we end up doing is trying to use the Bayesian networks to identify these for them. And we do a number of different types of gene selection and then build networks out of our selected genes. And I'm going to give you an example here in ovarian cancer. This work was done by my postdoc, Antonis, where we had different cell lines. The OV cell line was sensitive to cancer drugs, whereas the HOX was resistant. And then we had some treatment regimes where we gave them drugs and controls where they didn't have it. And then we looked at differential gene expression between these two treatments. And this slide just shows you that, it's true, the OVs, when you gave them drugs, this is mean relative tumor volume, they didn't tend to grow, whereas the control growed, but the HOX, it grew slower, but the drugged samples didn't actually have any reduction in growth. And then this is the type of standard bioinformatics we did on that. We did some clustering of the paths of these differentially expressed genes and identified them. And you might see here some of these clusters actually look like they're more than one cluster. I'll come back to that in a little bit. And then we do things like we use pathway over representation analysis to find pathways that are overrepresented in these different clusters, and then we hand those over to our biological collaborators who find that really interesting and want to go study those pathways. But showing what we did with Bayesian networks, actually worked with a visualization student, Thanasis, who's done some really cool stuff with me. But one of the things he did was develop a clustering tool where we could do hierarchical clustering with multiple different levels instead of cutting a clustering tree at one level where sometimes some of the clusters looked like they were more than one, we could sub-select them. And then I had an undergraduate student who used this to develop some clusters. Then, based on the original data that went into the differential expression, not the differential expression numbers, built some Bayesian networks and put in a variable that meant, was this sample treated or was it in a control? And then we were able to say, so we'd be getting kind of the most direct influence of what clusters of genes are most related to that treatment. And here, I do note this was an undergraduate thesis, and we did some somewhat silly things, but we still found some cool stuff. So there's the treatment node, and we found a number of clusters which are related to it. We probably should not have thrown in the OV and the HOX clusters together, because some of those probably have repeated genes in them. But even so, that would have made it harder for us to find an answer. And then when we took these genes, we could take them into independent clinical data set and split the data, patients, based on the expression levels of these genes. And we were able to find significant results of patients either taking less time or longer to relapse with their symptoms or to pass away based on the genes that she identified as being most directly related to treatment. And this is, once again, something our collaborators then want to go ahead with that list of genes. And just here is kind of, we do a lot of this now, just kind of finding groups of genes. This is identifying some genes related to alcoholism in rats. This is finding genes related to patients in melanoma. Here is one where we actually were after the pathways. We were trying to figure out, and this is actually protein phosphorylation data instead, so it's actually another level in that proteins, whether or not the protein can do something or not do something, and we were measuring that. And this was showing us which ones were being activated during treatment. And so now onto the ecological networks. So the idea here is what we're looking at is different species interacting with each other in a whole bunch of different ways. So they can eat each other, they can compete for the same resources, they can parasitize each other, they can have mutualistic reactions, and kind of the combination of these interactions are what we're calling our ecological interaction network. And here, I'll give a quick, I think I've got time for a quick example of my stuff before I pass over to Edwin, where we have data that we collected in bird groups in the Peak District National Park that were measured across two samples that were 14 years apart, and they did another one in 2018, 14 years later. It's a strange sampling interval, but they've done that, and we had different spatial scales that the data were collapsed over, and we developed Bayesian networks from this. And one of the things here, so this is just what the type of thing we did. This was kind of our control. This was the known habitat, and this is actually my favorite link here, which is altitude to path. So here, red is a negative influence score, blue is a positive influence score, and black, the influence score that Jing developed, can actually say, I give up, it's non-monotonic. And it does that because paths in the park tend to follow peaks or valleys. And it was very nice to see that the Bayesian network pulled that out. And then looking at the interactions among everything, including the birds, we found that the interspecific interactions were mostly positive, which was expected in bird communities. And when we looked at new species that moved into the park, we were able to find how they integrated into the network, and they did integrate kind of quite like they had been there before. They didn't have any kind of particular signature of being new animals. We found a particularly highly connected species, which was the golden plover, which is of interest. And also we looked at the spatial scale for interactions, and it turned out that raptors tended to interact with other species more on the larger spatial scales. And the raptors are the birds of prey, like the hawks and the owls and things like that, which made sense because they would have this kind of wider ranging distance that they would go to get their prey. And I think, yep, that's time for me to pop over, so I'm not going to get to tell you about some of the stuff we did, which was kind of incorporating spatial data into the algorithms as well. So if you want to know about that, you can talk to me later, and I'm going to pop over to Edwin's stuff. Yeah, there we go.
All right, so I'm going to start off my bit with a little bit of an introduction into ecological resilience, which is basically what I'm studying, and then I'll talk about where I'm actually carrying out my experiments, which Hans said it's on the rocky shores. Then I'll talk about why Bayesian networks are irrelevant, and of course my results. So, ecological resilience. You can actually just picture this as a ball that exists within a valley, and at the bottom of the valley, that confers a stable state. And the stable state confers a set of ecological relationships between species within a community, and that exists in equilibrium. Now, generally speaking, these systems can resist some sort of disturbances. So these disturbances might include something like a rise in temperature or any sort of physical disturbance event that might lead to a change in population levels within this community. Now, this disturbance can be visualized by the ball being moved slightly up either way of the valley, but generally speaking, if it's not too much of a disturbance, it's going to fall back, roll around, and it's going to stop at the bottom, and it's returned to an equilibrium state. Now, obviously, that's not always the case. Sometimes there's a lot of disturbance, sometimes it'll pass the threshold. And once that happens, it's going to fall into a new stable state, and this new state is going to be indicative of a new set of ecological relationships and a new network structure. And generally speaking, once you're in this new state, it's extremely hard to return to the original state, even if you're hypothetically able to remove whatever caused the change in the first place. So to gain a more intuitive sense as to how this works in ecology, here's a coral reef community. I'm sure everyone has seen this before. Generally speaking, corals, they provide habitat and shelter for thousands of species of fish, and also they exist in a mutualistic relationship with algae, where algae hangs out on the coral, provides it food through photosynthesis. But obviously there's now global warming, water temperatures increasing. Now as this increases, that can again be visualized by the ball being pushed up the valley. And if that crosses a threshold, then we have things like coral bleaching. That's when the coral starts to expel all of the algae, it loses its food source, it's more likely to die. And once it's dead, again, obviously that's practically impossible to return to its original state, even if the temperature were to hypothetically drop. So it's really important now to be able to understand the dynamics that regulate ecological resilience. What are the dynamics? Can we capture the dynamics of a system that's reaching a tipping point? And that's basically the scope of my research right now. And I'll be doing this on the rocky shore. And this is an example of a rocky shore. That's actually where I got my data. And rocky shores are actually really good to study these kind of things because they're generally a community on a rocky shore. They're very species poor. At best, you'd only have, within a community, at best you'd have maybe 5 to 6 interacting species, but the level of interactions between these species are very, very rich. So it's going to be extremely easy to experimentally manipulate to study ecological resilience. And also the most important thing is that it's been shown that there's alternative stable states on the rocky shore. So on one side, you see that it's on the left-hand side, that's basically rock that's completely dominated by barnacles. On the other side, it's completely dominated by fucoid algae. Now, depending which one's dominant, it's going to confer a completely different set of ecological relationships, and it confers a different set of species that's going to be present. So, for example, if fucoid is dominant, you're more likely to have snails and limpets that's hanging out underneath them, because it provides them shelter, provides them food, and also protection from predators. Now, how exactly does this translate into Bayesian networks? So here's a very, very simple community that you might find on a rocky shore. On the bottom, the dogwalks and the starfish, those are predators. In the middle, you have the top-shelled limpet and the littorinids. Those are the grazers. And obviously, the predators will prey on the grazers. And on top, you have the, what's that? That's the algae. That's the microalgae that hangs out on the surface of the rock. So if we know that, that the predators are present, you're reasonably certain to know that the grazers might not be there might not be too many grazers hanging around because they have to avoid the predators, or they might be dead. Now, if we know that the grazers aren't present, you're reasonably certain to know that the biofilm is going to be present because there's nothing that's going to be grazing on the biofilm. So, long story short, the conditional dependencies translate very well into the ecological relationships within the community. So now I'm going to talk about a little bit of what I'm doing. So I've been using quadrats to sample ecological data along the rocky shores. On control quadrats, I would sample 10 of them and basically do nothing with them and count data over 15 days. On the other side, I would remove all of the fucoid algae and track the changes, count the data over 15 days. And the original goal was to reveal a dynamic Bayesian network of post-disturbance changes to a community structure. But unfortunately, we could not actually work with a dynamic Bayesian network because, first of all, it was only 15 days. And what we found was that 15 days just wasn't enough for the community itself to reorganize itself after a disturbance. So we ended up sticking with static networks. So here's a control network that we found. And it's working pretty well. We managed to review the fucoid and barnacle competitive interactions for space, because when you find an area that's covered in fucoid, it's almost impossible for there to be barnacle to be settled on that area, and vice versa. But of course, with a static Bayesian network, you can't really model feedback loops, which is why we wanted to do dynamic networks in the first place. And also, it managed to reveal a really, really strong Bayesian relationships between limpid and linterinids and microalgae. And these are all consistent with what we would expect from literature. Now, then, with the fucoid removal network, that's when it gets a little bit more tricky, because it seems to be following a kind of, it's kind of having a same structure with the control network, but a few nodes might have rearranged. And what we think is happening is that on the control network, we notice that fucoid is very useful at predicting the presence of a lot of other things. But on the other side, because we removed fucoid but only sampled for 15 days, it has not been enough time for the species to reorganize themselves and change it into a different structure. So there might be a case where it's kind of like a ghost effect of fucoids being present. All we did is remove the fucoid, but all the things that would have been there on the control condition would still be there, because 15 days was simply not enough time for it to reorganize itself. So that is what we have so far. And what I'm thinking of doing right now is to use a control condition and some data visualization to act as some sort of blueprint for future study. So on the left-hand side, we have basically the most important interaction, and the strongest one, as mentioned, was the fucoid and barnacle. And when we visualize the data points and applied clustering on it, you can see that the distinct clusters that seem to be dependent on whether or not there's barnacle and fucoid present. So to me, what it seems like is that for future study, it will be interesting to see what would happen if you were to remove fucoid and remove barnacle as two separate conditions and track it over probably, I would say, at least 3 months, and see how that changes. And we'd have to find out ways to control for seasonal variation over time as well. So if anyone has suggestions as to how to do a dynamic Bayesian network, I welcome all advice, because I'm still pretty new to Bayesian networks as well. So yeah, that's all I have for today. And yeah.
So that's just me. I'll just pop back up and say thank you to people. I'll thank particularly Rick and Ian, who are the ecologists, who are Edwin's co-supervisors and have been helping him with the fieldwork part, and then kind of just lots of people who have helped in all of the different areas we've worked in, of course, people who have given us funding.
Cool.